All right, let's jump right into today's explainer. We're exploring a seriously mind-bending question that comes straight out of Dr. Nadia al-Mohammadi's research. We're looking at whether the ancient acoustic frequencies found in Scripture can literally, physically interact with human DNA. It's an absolutely fascinating journey through our source material, bridging the gap between bioinformatics, ancient texts, and cellular biology.
So we are unraveling a massive mystery today. Just think about it for a second. Can the sound of a specific word actually physically alter your DNA? What's really surprising here is that we aren't just talking about, relaxing music making you feel a little less stressed out. No, we're talking about acoustic waves acting as a literal mechanical switch on your genetic code. But to understand how something like that is even theoretically possible, we first need to look at the underlying mechanism. And that means we've got to talk about epigenetics. Think of your DNA as the master instruction manual for your body, right? It's the hardware. Well, epigenetics is the software. Through certain chemical processes, environmental stimuli, things like your diet, your stress levels, or in this specific case, sound, can actually change your gene expression. They don't rewrite the underlying code of your DNA, but rather they act like biological volume dials, literally turning certain genes up or turning them down. Now, what makes this source material so incredibly compelling is the clash between our two lead researchers. On one side, we have Dr. Nadia al-Mohammadi. She's a bioinformatician who hypothesizes that the acoustic properties of the Quran, specifically the word shifa, which means cure, leave a literal epigenetic signature on our DNA. And on the flip side, we have Dr. Helga Steiner, who is a strict materialist molecular biologist. Helga's intense skepticism serves as this perfect scientific foil as the two of them head right into their first major experiment. So, Nadia decides to test this out by exposing human skin cells to different audio environments for 72 hours. The groups that were listening to white noise or Mozart, they just showed random, fluctuating DNA methylation. But the group exposed to specific Quranic recitations, the data revealed a highly targeted 30% reduction in methylation across 20 specific genes. And crucially, these included FOXO3, which is widely known as the longevity gene, along with genes that regulate brain growth and inflammation. But predictably, Dr. Helga wasn't buying it for a second. She dismissed the findings entirely, basically chalking the whole thing up to a simple temperature fluctuation in the lab. And honestly, this perfectly highlights the rigorous burden of proof required in science. A 30% targeted reduction is a massive anomaly, for sure. But without a proven mechanism, a stubborn skeptic like Helga is just going to call it experimental noise. Nadia knew she had to dig deeper. To prove this wasn't just some random fluke, Nadia did something absolutely brilliant. She created a data-to-sound pipeline. She calculated the exact methylation differences in those 20 genes, converted them into a binary code, and then ran that through an audio sonification process. Basically, she literally wanted to hear what a biological cellular change sounded like. And the output from that sonification is where the research gets genuinely eerie. When played back, it didn't just produce a bunch of random noise. The sonified data of those genetic changes naturally produced a sound that matched 99.9% with the Arabic word shifa, meaning cure. It precisely replicated the phonetic structure. You had the hiss of the she, the explosion of the fa, and then the sudden stop. The DNA was echoing the exact sound that triggered its change in the first place. Confronted with this incredibly bizarre synergy, Dr. Helga raises the stakes. She proposes creating a completely synthetic DNA strand, one with identical chemical sites but completely devoid of actual human genes. Her logic makes total sense here. If this is some universal physical rule of sound-altering chemicals, well, it should work on synthetic DNA too. She promises that if Shifa comes out again, she'll finally believe it. Well, the synthetic test failed completely, yielding nothing but random hissing. But why? Nadia explains that while Helga copied the chemicals perfectly, she didn't copy the highly specific structural architecture found in actual human DNA. It turns out this biological phenomenon isn't just about the chemicals. It's about the literal physical shape of the human genome reacting to the specific 5,000 Hz vibration of the recitation. From there, the research moved to a second mechanism, histone acetylation, which dictates how accessible our genes actually are. The data showed a massive, targeted 40% spike specifically around anti-inflammatory genes. And Nadia found that this precise spike perfectly aligned with the rapid tapping phonetic pattern of the Arabic letter R in the word rahma, which means mercy. It was yet another perfect acoustic to biological match. And things got even more startling when they applied this to cancer cells. According to the source material, oncogenes, which are the genes that promote cancer, saw an increase in methylation, effectively turning them off. Conversely, the tumor suppressor genes were activated. This epigenetic reaction matched the phonetic friction of the Arabic word kasara, meaning loss for the unjust. The sound was acting as a targeted detriment specifically to those cancer cells.
This brings us to a huge, pivotal turning point in the narrative. Dr. Helga, our steadfast skeptic, takes it upon herself to test the cancer data across 5 entirely different tumor lines, every single time that phonetic friction pattern returned. Confronted with the undeniable replication of the data, she finally concedes. Within the context of the research, the biological reactions to these specific words are genuinely coded right into the cells. Now, to move from pure theory to empirical evidence, Nadia launched a clinical trial with 50 patients suffering from chronic fatigue. After 3 months of daily audio exposure, FOXO3 longevity methylation dropped by 25%, while brain growth expression increased by 40%. But most importantly, the real-world clinical fatigue scores improved by 30%, a result completely absent in the placebo group. Nadia termed this whole process epigenetic music therapy.
What I find incredibly ironic here is the complete role reversal of our characters. Armed with this empirical data, it is actually Dr. Helga, the formerly stubborn materialist, who steps up to officially present the clinical trial data to the Institutional Review Board. She becomes the primary scientific advocate, cementing the idea that this is a reproducible, medically significant phenomenon. The real climax of the source narrative occurs at an international genetics conference. Nadia stands before the global scientific community and visually proves her point. She projects a heat map showing the physical methylation differences in the cells and perfectly overlaps it with the acoustic audio envelope of Surah al-Isra. The visual alignment is an astonishing 99.9% match. The dips, the peaks, and the stops in the audio perfectly mirror the turning of those biological volume dials.
This is where the science gets incredibly granular. The mapping showed that each specific acoustic sound acted as a distinct biological key. For example, the 5,000 Hz hiss of an S sound triggered one specific gene to unmethylate, while the 2,000 Hz explosion of an F sound triggered a completely different anti-inflammatory response. The sounds were fitting perfectly into cellular locks.
Following this super detailed breakdown, Dr. Helga offers a highly dramatic public endorsement. She admits to her peers that, based on their replicated data, she now believes the acoustic properties of the scripture act as a shifa, a cure, mechanically on the DNA itself. Her narrative arc from a skeptic dismissing temperature fluctuations to a convinced advocate is fully complete.
So, what does all this mean for the future? Well, according to the source material, this research is already pivoting toward clinical applications through the Shifa Gene Project. The laboratory is currently testing how combining specific recitations can actually modulate stem cells, effectively accelerating the body's natural ability to repair tissue. It's an entirely new frontier of non-invasive therapy.
Nadia's philosophy ultimately boils down to this one thought: don't just feel the cure, feel the methyl. It's a really powerful reminder from this research that our environment, right down to the specific acoustic frequencies we listen to, might be actively programming our biology. If our DNA is quite literally reacting to the structure of sounds, how might we rethink the power of language and belief entirely? Thanks for joining me on today's Explainer. Keep learning.